A very good afternoon, dear students. Welcome back to the class. Hope you all have studied and learned about carbohydrate metabolism. Now let's move on to biological oxidation in the same unit. The next title under unit 2 is biological oxidation. In that part 1 we are going to discuss today. The contents are electron transport chain ETC and its mechanism. What is the title? Electron transport chain ETC and its mechanism. Let's see about electron transport chain, that is ETC. What is an ETC? The byproducts like NADH and FADH2 are released from catabolic processes like glycolysis, Krebs cycle, beta oxidation, etc. And these compounds will get oxidized through ETC, electron transport chain, to release the energy as ATP. ATP is released through the process called chemiosmotic phosphorylation. Chemiosmotic phosphorylation, osmosis process due to chemical transport. So it is called chemiosmotic phosphorylation. And how ATP is synthesized? From NADH and FADH2 through electron transport chain. When electron is transported, the process is called maybe redox reaction. Reduction and oxidation takes place when electron is transported. Loss of electron is called oxidation, and gaining of electron is called reduction. So this is how the electron transport chain forms to release ATP. What is the theory behind electron transport chain? It is to produce ATP, a high energy molecule. How? From driving ATP synthase. How ATP synthase is driven? By increased concentration of hydrogen ions. Where it is increased? In the intermembrane space of mitochondria than in the matrix, mitochondrial matrix. How it is increased? How the concentration of hydrogen ions are increased? By means of producing hydrogen ions through ETC at 3 specific points in the chain. How it is increased? By passing electrons from one membrane bound electron carrier to another, one carrier to another carrier. Where these electrons are transported from the byproducts of catabolic processes. What are the catabolic processes? Glycolysis, TCA cycle, beta oxidation, etc. And what form electrons are transported? As hydride ions. What are hydride ions? They are present along with the coenzymes called NADH or FADH2. And these hydride ions will be carrying electron from one carrier to another carrier. Thereby hydrogen ions are released from the electron transport chain, and that increases its concentration, and which drives ATP synthesis for releasing ATP from the electron transport chain. This is the actual theory behind ETC. What are the electron carriers? There are 4 types of carriers present in electron transport chain. They are named with Roman number 1, 2, 3 and 4. And the first one is NADH ubiquinone oxidoreductase. Second one is succinate ubiquinone oxidoreductase. Third one is ubiquinol cytochrome C oxidoreductase. And fourth one is cytochrome C oxidase. Cytochrome C oxidase is a hydrophilic carrier, where Q, ubiquinone or ubiquinol, it is an intermediate electron carriers. Q represents ubiquinone or ubiquinol, it is a hydrophobic carrier. Whereas other ETF and glycerol 3-phosphate dehydrogenase also electron carriers. This electron flow causes oxygen synthesis and proton motive force causes ATP synthesis. Since proton motive force activates ATP synthesis for releasing ATP from the electron transport chain. Let's move on to the mechanism of electron transport chain, that is ETC. Here you can find 4 different formats of electron transport chain. In the coming slides, you can observe whichever format you understand better, and all the 4 formats shows same mechanism of electron transport chain. Where this electron transport chain is located? In mitochondria. Which part of mitochondria? Inner membrane of mitochondria. Inner membrane of mitochondria. So mitochondria has outer and inner membrane, and this electron transport chain is located in the inner membrane. You can observe this chain has 4 complexes with the name is mentioned near to the each complex. And what is the reaction actually happens there, and each complex releases protons, that is hydrogen ions. When hydrogen ion release occurs, electron is transported through the chain. When highest concentration of hydrogen ion represents the release of ATP in the mitochondria. We have seen 3 points hydrogen ion is released. You can observe at complex 1, complex 3 and complex 4 hydrogen ion is released. Thereby in the intermembrane space hydrogen ion concentration is increased. Because of this increased concentration, ATP synthase is activated for synthesizing ATP from ADP. Here is the format 2 for mechanism of electron transport chain, and this electron transport chain is located in inner membrane of mitochondria. Between intermembrane space and mitochondrial matrix, ETC is located in mitochondria, which has 4 complexes to carry electrons and ATP synthase enzyme. Among 4 complexes, 3 complexes are releasing hydrogen ion in the intermembrane space. This increases hydrogen ion concentration in the intermembrane space. This causes activation of ATP synthase and this synthesizing ATP from ADP through phosphorylation process. The final product is water molecule through ATC reaction. Here is the format 3 and 4 for mechanism of ETC, and these 2 diagrams represents mitochondria with outer membrane, intermembrane space and inner membrane, and outside of the mitochondria you can find cytosol also. And where ETC is located here, you can see clearly it is in inner membrane. Where hydrogen ion is released? In intermembrane space, that is between outer membrane and inner membrane. From where you are getting NADH and other byproducts from mitochondrial matrix through Krebs cycle, and the released byproducts are involved in ETC chain for synthesizing ATP. ATP is synthesized because of increased hydrogen ion concentration in the intermembrane space, so that ATP synthesis is activated for releasing ATP as energy molecule, high energy molecule. Actual mechanism of ETC involves electron flow and proton motive force. Where electron flow through the ETC causes oxygen release and proton motive force causes ATP synthesis. Oxygen release and ATP synthesis are the role of ETC. Electron flow and proton motive force are mechanism of ETC. Clear? So with this we are coming to the end of discussion on electron transport chain and its mechanism. Hope you all have understood this topic. Thank you all for patient listening. In the next class we'll be discussing about part 2 of biological oxidation. Thank you all.